Hi everyone, today we will be dissecting a cow eye to analyze the internal structures. To perform this lab, you will need a lab coat, gloves, a scalpel, a scissors, and a tweezer. To begin, we will start by removing the fat around the eyeball. This is beneficial to expose the sclera and the optic nerve. This ensures a clear view of the eyeball structure, allows for proper handling, and makes it easier to accurately cut into the eye for examining internal components. Upon removing the fat, you will begin to see the extraocular muscles. Ensure to remove those as well. Upon the removal of the extraocular muscles, they were measured and one of them was found to have a length of 40 millimeters. Now that the fat and the muscles have been removed, we are now able to identify the sclera and the optic nerve clearly. We will now measure the horizontal visible iris diameter, vertical visible iris diameter, and the axial length of the eye. On humans, this step is beneficial for accurate contact lens fitting, monitoring eye growth, and managing myopia progression. Axial length is the primary indicator of myopia progression, with 1 millimeter of growth resulting in a 3 diopter shift. Now that we have completed our measurements, we will now divide the eyeball into its anterior and posterior halves. To do this, we will use the scalpel to make a small incision into the sclera and then proceed to use the scissors to cut around the circumference of the eye, ensuring not to penetrate the internal components of the globe. As you separate the posterior and anterior halves, you will begin to notice that the aqueous humor is leaking out of the eyeball. The aqueous humor is located between the cornea and the lens, and it serves to maintain intraocular pressure, provide nutrients, and maintain proper refractive index. The eyeball has now been separated into its anterior and posterior halves, with the anterior half containing vitreous humor. As we begin to identify structures in the posterior region, we will first encounter the retina, which is a thin tissue-like material containing photoreceptor cells used to create electrochemical signals to the brain. Upon removing the retinal layer, you will realize that it is attached to a region of the eyeball. This is where it is connected to the optic nerve. Located under the retina is the choroid coat, which is a highly vascularized area providing the outer retina with oxygen and nutrients. It also contains a reflective coat called the tapetum lucidum. This structure reflects light back into the photoreceptors, significantly improving low light vision, causing the glowing effect known as eye shine. This feature is absent in humans as we are more active during the day, therefore excess light is absorbed to enhance vision clarity in photopic conditions. To begin analyzing the anterior region of the eye, we must first remove the vitreous humor. We can then observe that the lens is physically connected to the vitreous humor via the hyaloid fossa of the vitreous membrane. This connection stabilizes the lens and protects it from oxidative damage. However, we need to remove the lens to further analyze its function. Upon removing the lens and washing it with water, you can observe that it is spherical in shape. This serves to evenly refract light from all directions onto the retina. This uniform round shape acts as a biconvex magnifying lens, ensuring that light rays from the peripheral view are bent equally to meet a single focal point, preventing image distortion. We have then turned the anterior part of the eye inside out, and we are now observing the ciliary bodies as well as the iris. The iris serves to regulate the amount of light entering the eye and is located over the lens. The ciliary muscle controls the shape of the lens, known as a process called accommodation. This is done by contracting or relaxing the muscles depending on the distance of the object being viewed. We are now gently separating the iris from the cornea to prevent it from being cut when the cornea is being removed from the sclera. To remove the cornea, make a small incision at the limbus using a scalpel and then use a scissors to cut around the periphery of the cornea. Now that the cornea has been separated, we can observe its tensile strength and flexibility due to the organized arrangement of collagen fibers which serves to protect the eye. Note that a healthy cornea is typically clear to allow for the uninterrupted passage of light into the eye. We are now observing the iris after it has been removed from the sclera. This lab was conducted by Jayla Francois, Fatima Rock, Nayan Maharaj, and Brian Integrant. Thank you for watching.